Assalamu alaikum. How are you, students? Dear students, I have come before you with a new topic of chapter 14, Genetic Engineering. But do you know what is genetic engineering? Genetic engineering is the combination of two words, gene and engineering. Dear students, you have idea about gene. You know, gene is the part of DNA. What is DNA? DNA is a hereditary material found in the chromosome of nucleus of cell of organism. Gene, the part of DNA, controls all the characteristics of organism. Now, engineering. Engineering is the technique of work and using the tools and machineries skillfully. Now, genetic engineering is the engineering with gene, the technique through which recombinant DNA can be formed is called genetic engineering. Through genetic engineering, recombinant DNA is formed. Now, what is recombinant DNA? Recombinant DNA is a DNA molecule that has been modified to include genes from multiple sources. So, we can say that recombinant DNA is a modified DNA molecule with desired gene. Through genetic engineering, it has become possible to transfer the gene from one organism to another organism. We can transfer gene from a bacteria to a human being, a plant to an animal to a plant. The organism which is produced through recombinant DNA or modified DNA molecule is called genetically modified organism, genetically engineered or transgenic. So, through genetic engineering, recombinant DNA is formed, and you know what is recombinant DNA. Through genetic engineering, it has become possible to transfer the desired gene from one organism to another organism, and the organism which is produced through genetic engineering is called genetically modified organism or GMO, genetically engineered or GE or transgenic organism. Now, steps of genetic engineering or recombinant DNA technology. Step 1, isolation of DNA from two sources. Step 2, cutting of DNA with restriction enzyme. Step 3, adhesion of both DNA with DNA ligase enzyme. Step 4, insertion of recombinant DNA into bacteria. Step 5, cloning of bacteria. Now, step 1, isolation of DNA from two sources. That means DNA molecule from a carrier and DNA molecule from another sources are to be collected. Step 2. Cutting of DNA with restriction enzyme. After collecting the DNA from two sources, we have to cut the DNA with a special enzyme known as restriction enzyme. Step 3. Addition of both DNA with DNA ligase. After cutting the carrier DNA and desired DNA, we have to adhere the desired gene or DNA with vector DNA or carrier DNA with a special enzyme known as DNA ligase. Step 4. After adhere the two DNA molecule or two DNA, we will get a new DNA or modified DNA molecule which is called recombinant DNA. Then this recombinant DNA is to be inserted into a carrier bacteria. Step 5. Cloning of bacteria. You know, bacteria is able to self-reproduce. By the process of cloning, bacteria will increase its number, and as a result, the recombinant DNA with desired gene also increases its number, and this process is called cloning. Now, dear students, you will get clear idea about genetic engineering or recombinant DNA technology with this figure. It is a bacteria and it's a common bacteria. It is known as E. coli or Escherichia coli bacteria. This bacteria is found in the alimentary canal of human being. It is known as plasmid bacteria. Dear students, you can see a three circular structure inside the bacteria. These are known as plasmids. This plasmids is one kind of DNA, and this plasmid DNA is to be used as carrier. Now we will take an animal cell. We will transfer the gene of the animal cell to the DNA of E. coli. So first step, isolation of DNA from E. coli and animal cell. Now we will do that. Here we have isolated plasmid, that means DNA plasmid from bacteria. Now we will isolate the desired DNA or gene from animal cell. It is desired gene. Next, second step, cutting of DNA with restriction enzyme. Here are plasmid DNA and desired DNA or gene. Now we will cut some part of plasmid DNA and desired DNA with a special enzyme known as restriction enzyme. Now the third step, development of recombinant DNA, addition of both DNA with DNA ligase. We have got two parts of two DNA molecule. Now we will adhere these two parts with a special enzyme known as DNA ligase. So we have got a new DNA molecule, or that is modified DNA molecule with desired gene. This is desired gene. and this DNA molecule is called recombinant DNA plasmid. Now, fourth step, insertion of recombinant DNA into E. coli bacteria. We will insert the recombinant DNA plasmid into a new bacteria. It is also E. coli bacteria. So, this E. coli bacteria will be with a recombinant DNA plasmid. Next, fifth step, cloning. And you know, bacteria is able to multiplicate itself, so it will increase its number by the process of cloning, and this total process will be called recombinant DNA technology. I think, dear students, you have got clear idea about genetic engineering or recombinant DNA technology. If you have any problem, you will watch the video more than one time, then you will be clear about this topic. Thank you, students. Assalamu alaikum.